Hi, welcome to Consider This Simplifying the COVID-19 Conversation. I'm Marianne McDowell, and I co-host this series with Heidi Beidinger-Burnett. Well, thanks for joining us again. This week's session will be on vaccines and the immunology of COVID-19 with our guests, Dr. Brian Baker and Jeff Shorey. But before we would go into the live session, I thought I would give you a little tutorial on some of the immune mechanism terminology. So let's imagine a mild case of COVID-19 where an individual gets exposed and then the virus grows in the body, eventually dissipating. We call this the viral load. And the individual gets symptoms about 5 to 7 days after exposure and is infectious a few days before symptoms arise, all the way through till symptoms are recovered. And the individual will recover in about 2 weeks. So what's happening with the immune response? So we have two sides of the immune response: innate immunity and adaptive immunity. So the innate immune response is what happens first, and it gets enlarged and eventually dissipates. And the goal of the innate immune response is to get rid of the infection or the virus. But if it can't do this, then it activates the adaptive immune response much later. So what is an innate immune response? So in innate immunity, it's the first line of defense. So let's imagine we have our lungs and our SARS-CoV-2 virus comes in. I'm going to blow it up so we can look more closely. And the innate immune response has lots of preformed proteins and functions, as well as cells that get recruited to the site of infection. And the innate immune cells that we think of are dendritic cells, macrophages, and neutrophils. And these cells, when they're recruited to the site of infection, release lots of mediators, including cytokines. The job of these mediators is multifactorial. One is to recruit more cells like themselves. And oftentimes in infection, we think of neutrophils. And so when a physician is counting your white blood cells, they're usually counting for these neutrophils or innate immune cells. But all of these processes together end up causing inflammation, pain, redness, swelling, all of the things that you typically think of as inflammation. And what you might not typically think of is a function of inflammation is actually clotting. And certainly we know that's important for COVID-19, that clotting, or you might have heard the term thrombosis, or fibrinogen and fibrin, causes clots in a lot of the functions and decreased functions of the airways. A marker of this is D-dimer, and that's actually the breaking down of the clots, but oftentimes you'll see physicians reporting a D-dimer levels. So if the innate immune response cannot get rid of the virus, it actually activates adaptive immunity. And it does this by taking parts of the virus and presenting it on the cell surface, in particular dendritic cells. And then they migrate and activate T cells, cytotoxic T cells. And when they're activated, they divide, make more of themselves. And this cell type's job is to actually kill infected cells, the cells that are infected with SARS-CoV-2. These dendritic cells also activate T helper cells, which also proliferate. And T helper cells provide help to all parts of the immune system, but combined CTLs, or cytotoxic T cells, and T helper cells is what we call cellular immunity. T helper cells are also involved in activating B cells. So B cells have antibody on their surface. And if the antibody recognizes SARS-CoV-2, it takes it up and presents parts of it to the T helper cell. And then the T helper cell activates the B cell to change dramatically and proliferate or divide. And these differentiated B cells are called plasma cells. The role of plasma cells is just to release tons and tons of antibody out into the milieu, if you will. And these antibodies have different functions, one of which is to neutralize SARS-CoV-2 virus. But it turns out that we have different kinds of antibodies or classes of antibodies: IgM, IgE, IgA, and IgE. The Ig stands for immunoglobulin, which just means antibodies. IgM is actually 5 different antibodies bound together. IgG and IgE are monomers, and IgA is 2 bound together. Now, these different classes have different functions and different distributions. IgM always happens early in an adaptive immune response. And it isn't until later that these other classes come up and arise. IgM can be found in the blood, some of the tissues, and the mucosa. Importantly, IgG is found in the tissues, IgA in the mucosa. So what I mean by mucosa are like your epithelial lining and in your nasal passages. So you can imagine if you really wanted to block SARS-CoV-2 infection, you'd want to have a lot of IgA where the virus gains entry. IgE is actually found in the skin. As IgM is activated early, it actually turns on some of the innate immune response functions. IgE isn't very important in COVID for COVID-19 as far as I know, but it is important for allergy. So some of you might be having some seasonal allergies, and IgE actually makes some innate immune cells release histamine. IgG and IgA are most important for neutralization. So I'm going to give you a little animation here. Imagine the cell surface of an epithelial cell. It has this receptor ACE2 on the surface, and here's our IgA. So the SARS-CoV-2 gains entry into a cell via ACE2, but these neutralizing antibodies block that, and so the virus cannot gain entry. IgE, or IgG rather, has another possible function called ADE, or antibody-dependent enhancement. We don't know if ADE is happening with COVID-19, but we know it can happen with other viral infections. So what ADE is some cells have receptors on their surface that can recognize antibodies themselves. And what happens is then the SARS-CoV-2 is recognized by the antibody, and then the Fc receptor recognizes the antibody, and it actually lets the virus get into the cell. So that's the basis of immunity, especially with a mild case of SARS-CoV-2. But what happens if the innate immune response actually does control the viral load? Well, oftentimes there might be very little or few or no symptoms, and the adaptive immune system may not get activated very highly at all. What's the opposite? If the innate immune system can't control the viral load at all, what happens in that particular case is the innate immune system stays active, and the adaptive immune response stays active in its much higher levels. So all those mechanisms that we talked about in the case of innate immunity, as well as adaptive immunity, are happening at huge levels, causing a lot of damage to the tissues, and that's what leads to the severe pathology. So that's all I have for now. If you have any questions, please email us at consider@nd.edu. Thanks again, and I'll see you real soon.